Affinity Chromatography. What is it, how does it work, and why is it useful? Affinity chromatography is a separation method based on specific binding interactions between an immobilized ligand and its binding partner, such as an antibody-antigen complex, an enzyme-substrate complex, or an enzyme-inhibitor complex, and the interactions between these two. A nice analogy for this phenomenon is that of a key which specifically fits to a certain lock. The important point in this analogy is that the ligand captures the wanted protein, making it stick to it, the ligand being the lock and the specific protein being the key. The degree of the purification varies but can be quite high depending on specificity of the interaction used. The workflow can be divided into 5 major steps. First, cell lysate, or our sample, is added to the column containing both wanted and unwanted proteins. Second, the protein of interest will interact with the beads through matching affinity. Third, we add a wash buffer and remove remaining unspecific protein and other substances, until all that's left is our wanted protein, hopefully at least. Fourth, we add an elution buffer to elute the protein of interest from the affinity beads, making sure that our protein disconnects from these beads and gets removed as well. This can be done through pH change or a change in salt concentration. These changes in the environment is what causes our protein of interest to unbind from the ligand. Fifth and finally, we have our purified and active protein of interest that can be collected from the column. So why is affinity chromatography so useful? Well, first of all, it has a high selectivity, meaning that it is excellent at distinguishing between different proteins. It also has a high resolution, meaning that there is a wide separation between the peaks in the results, allowing for easier analysis of the different compounds present in the sample. Finally, it has a high capacity, meaning that the ligand is able to retain the protein of interest for a long time, allowing for easier and nicer separation between the different compounds in the sample. Affinity chromatography is a very common first step in protein purification and is often followed by size exclusion chromatography, which coincidentally should be displayed on the screen right now. So please check that out if you want to know more about protein purification and these different steps that are usually taken to ensure that our proteins are purified after we have made them. And as always, thanks for watching. Until next time.